Good morning, everyone. Happy Friday. If everybody can see in the group chat, we actually would love to hear where everybody is dialing in from. I think it's cool to see people coming from all over the country as we are at Viome, scattered about. Thanks for joining today's call. I think everybody knows me, but I'm Rob. Hello, I'm one of the account managers here with Viome. And we've got a pretty good agenda for you today that I'm certainly excited about. First, just one quick update. You guys may have noticed that we have a new portal for the ambassadors. So there's some new features in there. We've cleaned up a lot of things. Frankly, we think it looks better, more visibly pleasing. So one of the big things that you guys have asked for since the beginning of the year is more resources. And that relates to marketing resources, logos, banners, photos, those sorts of things, materials for education, so blog posts and links to articles and the ability to share all of that with your following, and then also some videos. And so we've put in an asset library where all of that stuff is available. There's some great content in there right now. There's going to continue to be more as we build that out over the next 6 weeks or so. And you guys have now access to all of that stuff without having to go through us as you guys build out your personalized marketing. The other thing that we did is we cleaned up the sales and commission tracking and just how that's visible for you guys. So we tried to make that more intuitive, more clear in terms of payouts and tracking individual commissions and all of those sorts of things. So hopefully that's helpful for you guys. If you guys have questions already on that, Lochum here with our product management is going to be on the call to answer some of those questions. And if you guys haven't checked out the portal, give it a spin sometime in the next few weeks and let us know what works and what doesn't work, because that's how we are able to improve upon this stuff. So today's speaker is Lisa McClellan. She is our CLIA General Lab Supervisor. She works at the laboratory in Los Alamos. And the reason we wanted to have her on today is to talk about the journey that the sample takes as it goes through the sequencing process. Because one of the questions we get is, why is it that it takes 4 weeks for me to receive my results and recommendations? And the reality is because this process is so sophisticated, and it's, we are the only people, we're the only organization that can do this, that can look at RNA. And so what I thought would be helpful is to have her walk through that process that your sample goes through and how the sequencing works. So Lisa has spent the last 3 years at the Los Alamos National Laboratory researching tuberculosis protein structures as well as algal biofuels. So I can't pronounce that. So she's smart when she's doing those sorts of things. And then obviously she works with us on all of the sequencing aspect of our kits. So before I turn it over to Lisa, just a quick reminder that we're going to keep everybody on mute until the last 10 minutes of the call. But if you guys have questions during the call, please put those into the chat, and Erica or Ashley or one of the lovely support people can either answer that or keep it until the end and we can expand upon it. So with that, I'm going to turn it over to Lisa. Lisa, good morning.
Good morning. We're here in Los Alamos at the Viome lab here.
Great. So you're going to walk us through the full kind of workflow of the sequencing, and then we'll expand upon some of those individual steps and help give people a better sense of the science, why we're unique, and why this process is 4 weeks. And because the reality is there's really no quicker way to do this.
Right. Okay. So here's like an overview of our Viome laboratory workflow. I know this slide is a lot, but I'm going to go ahead and talk about these first sequences here in red. So one of the first things when the sample gets into the building is we're going to receive the order. So that goes through specimen accessioning first, and I'm going to go through more of these processes later on. And then we're going to extract all the RNA from the samples, and we're going to run a DNA treatment, quantify the RNA that's in the samples that we received, get rid of the rRNA through our RNR process, run another DNA treatment, ddNA synthesis, DNA size selection, PCR amplification, library pooling and cleanup, and library QC. So this step already takes a few days.
And Lisa, I'm going to actually have you go back quickly just to add some context for people. So as a reminder, we are sequencing RNA, not DNA, which is what every other company is working with. And for that reason, if you were to look at a 16S or a metagenomic workflow that it was broken down like this, it would be significantly shorter. There would be a lot less steps. And again, when you're looking at DNA, you're not looking at functionality. You're not, in this case, looking at what the bugs are doing. You're simply looking at what's there. So it's a much lower resolution and much less actionable review of the microbes. Okay, so just wanted to share that. So I'll turn it back over to you.
Yes, thanks. So specimen exceptioning. So we have our lovely lab assistants here. So when the specimens come through the door, the first thing they're done is unpackaged, and then the specimens are scanned into our LIMS system, and then all aliquoted into smaller tubes for processing and storage. Then after accessioning, then it gets moved onto our automation system. So I have a video here of how we use Hamilton Star systems here. And here's a video of how it works. So a lot of this is just liquid handling that it performs.
So Lisa, the Hamilton machinery here, is this standard for the industry? Is this unique?
No, it's much more unique, and it takes away any sort of operator error or human error that can go into the process. So all these are programmed to be exactly the same and treat all the samples exactly the same. So as you can see here, it's taking the sample over here and making sure that everything gets put into the wells. So you reduce any chance of human error by, say, missing a well.
Okay, so the first
automated system.
So the first two slides that you touched on were basically getting the sample at the lab, properly putting it into our system so we can track it, and then putting it in a structure where then this machine can accurately set up the sequencing process, meaning put it into these wells where the ultimate sequencing is going to occur. Is that fair?
Correct.
Okay.
So the first thing we're going to do is take our stool samples and all the microbiome in there and extract the RNA. So the first step is manual lysis and chemical lysis to break up the cell walls. So you can see it here, it broke up the cell walls, the samples, and then it's going to get rid of the DNA. And so all we're left with here is the RNA. So here's all our RNA, and we're going to get rid of the ribosomal RNA because we're specifically going to be looking for mRNA.
Which stands for messenger RNA?
Yes. So here we're left with our mRNA, and we're going to do another DNA treatment to make sure that all the DNA is removed.
Beautiful. So if I'm understanding this correctly, you start with, basic cells in the sample, and in order to get, and these are the cells of the microbes, and in order to get to their RNA, you have to break the cell apart, and that's the lysing portion of that, if I have that right. Okay. And so lysing is important because obviously you don't want to damage anything in the cell, but you do need to break it apart enough to be able to see inside. And then once you get past that step, if I heard you correctly, then you have two sets of information. You have DNA and you have RNA mixed together. And somehow we need to strip out the DNA and isolate just the RNA before we can even start to look at functionality and those sorts of things. And then actually, and we didn't chat about this yesterday, but then you have ribosomal RNA and then mRNA, and then you need to separate those two out as well because it's the messenger RNA that is really
that's carrying the genetic code that we're looking at for everything that's metabolically active.
Perfect. Okay. All right. So I'm with you.
Okay. So the DNA treatment is, used with a DNA enzyme that will strip all the DNA out and just leave us with the mRNA.
And that's, by the way, this video is showing that taking place as we move through the steps.
Correct, yeah.
Okay.
So then the next process would be to create cDNA, complementary DNA from our mRNA. So here you can see us making copies of our RNA into cDNA.
And why is that important?
Because we're going to sequence the cDNA.
Okay, so to phrase that another way, the cDNA is much more legible as we sequence than just the raw mRNA.
Yes.
Okay.
And it's much more stable because we ship it out to a reference lab for sequencing. So it'll be stable during shipping.
Ah, okay.
So then the next step that's performed is size selection to select for certain sizes, base pair sizes of our cDNA. And then after that, we will perform PCR amplification in order to make multiple, like hundreds and hundreds of copies of those same cDNA sequences.
So we've basically now distilled, we've been distilling, almost like you would with like, vodka or some sort of liquor, where you're trying to get rid of impurities, trying to get rid of confusion to get to RNA. Then you need to convert it into a way that is actually legible as we go through the sequencing process. And then once we get there, we want to be able to clearly see what's going on, which would be the equivalent of cranking the font from 10 to 50 or 100.
Right.
Making it really large so that we can see. But instead of making the actual RNA large, it's quantity. Is that fair?
Yes.
Okay.
So then we're going to pull the libraries together, and then we are going to send it out for sequencing. That's the only portion that is not done in the lab here. So our sequencing and bioinformatics sequencing is performed either at UPMC or OMRF. UPMC is UPenn, I believe, in Pittsburgh, and OMRF is in Oklahoma. Then once our sequencing data is returned, it undergoes the bioinformatics pipeline. And what the bioinformatics does is map out all the species and strains of all metabolically active bacteria, viruses, yeast, fungi, parasites, and bacteriophages. So then this information is sent on to the translational science team and interpreted into your results and recommendations for all our patients.
Mm-hmm. So from Viome's standpoint, what really makes us unique was all of that proprietary distillation down to the RNA, making it functional, so to speak, from a legibility standpoint, and then sequencing it at one of these two facilities after the fact. And then because of all the prep work that you just described going into that, it gives us the ability to then go back and look during the bioinformatics at the species and the strain. So once we see the functionality so clearly, we can go back and see which bugs specifically at strain level were producing these. Is that fair?
Right. So that's part of this paper actually that was just accepted yesterday into the International Journal of Genomics that has a lot of the data that supports the science behind our metatranscriptomic technology, as opposed to 16S, which only reads at the genus level. And just wanted to touch on some of the other work that we do here at the Los Alamos Lab. We just launched our food sensitivity intelligence test. And we are also working on the GXI gene expression test. And we also perform a lot of reagent preparation on site, as well as research and development and all the studies.
That's a lot of stuff going on at that lab. Very cool. And the FSI, the GXI, because they're different tests, don't go through this process that you just described with us, or is it somewhat similar?
Completely different. So food sensitivity intelligence, we run an ELISA assay on IgG. So it determines what foods you're sensitive based on your blood serum. Actually. Mm-hmm. So it's a complete different sample and gives you
complementary results.
Yes.
Okay, perfect.
And gene expression test is developed, still in development right now, and it's based on blood. in what genes are expressed in your blood.
Mm-hmm. Which would be similar to a lot of the other DNA tests that are out there, but this would be an RNA test based on blood.
Exactly. So it'd be what's metabolically active.
Terrific. So Lisa, I have one quick question for you. In terms of the full process, cradle to grave of the sequencing, or of us getting the sample and then going through the sequencing, is there anything specifically that might delay the process or a particular step in the process or elongate anything in particular?
Rob, there's a few things that would elongate the process. One is we send samples to our sequencing facility. We have to get enough samples first to send to the sequencing facility, which is, we send out 752 samples at once. So usually it takes us about a week to gather that many and process that many. And another thing that could delay the process is also waiting for signatures for certain states. The CLIA requires additional states to have authorized signatures through the portal, and that can sometimes take up to another week. Another thing that could delay the process is it takes about a week to get sequencing data back from our reference labs.
Okay, so to recap, because I know that in the presentation you mentioned that once we go through the distillation down to RNA, then we send out that information to Pittsburgh and Oklahoma. In order, before we can do that, because we're not going to send out, each kit or each sample individually, that, is not cost effective or time effective, we have to wait until we reach critical mass. That critical mass is over 700 samples. And so if we don't reach that, then we simply can't send it out. And that takes about a week to reach that point. And then the next thing that could delay it, if I heard that right, and folks should be familiar with CLIA at this point, but us being a clinical lab at this point, not a research lab, we now require doctor sign-off in specific states for the samples to be processed. And that is on an individualized basis, so that can take some time, so depending on which state you're in. And then give me the third one again.
Sequencing data takes a week to get back from the sequencing reference labs.
Okay, so once we have completed those first two steps, then it's going to take a week regardless to come back from the sequencing lab. And that's out of our control because that is, at this point, a third party.
Right.
Okay, cool. And that's the end.
Okay. Yep.
I love it. So yeah, just to quickly recap here, the significance of being able to interpret RNA is something that can't be done anywhere else. And it's only through this process that we're able to accomplish it. And so, when people say it'd be nice to have the results in a week, or, there's urine tests that come back in a day, it's impossible to be better, cheaper, and faster. You can't have all three. And
Right.
So to have this resolution in this unique of a test, in this meaningful of results, takes more time and is a more labor-intensive process. But I think you did a tremendous job, Lisa, walking us through that. I know that I had no concept of how this functioned until we spoke yesterday. So I hope that it was helpful for people. And at this time, Ted, what I'd love to do is open up for questions. Oh, people are from all over.
While they're thinking of that, I like the analogy of musical instruments in a house, right? I have a piano and a guitar in my house, and that's like my DNA, I guess you could say, and there's people and bodies in there. But to actually have somebody who knows how to use the instruments and actually can make beautiful music, those are different levels and different steps. And then to actually take that music that someone produces and study every single note, that would be kind of like what we're doing versus what some other companies are doing.
Right.
I love that. That's a great analogy. And it incorporates the more important aspect of this, which is what can you do about it? So, those other tests that show you your genes, well, those are fixed entities, fixed concepts, whereas expression is malleable to some degree, and the ability to, through food recommendations and dietary changes, you can have an impact on that and move the system in a better direction, which is awesome.
And that's what's so great about the metatranscriptomic as compared to 16S, is that we're actually getting to the strain level and we can determine any kinds of recommendations from that. So
yeah, absolutely. Ted, have we Have we unmuted?
Yeah, everyone, I've made it so they can unmute themselves.
Okay. So if anybody has a question, feel free to unmute yourself and ask. And as a reminder, if you guys have any questions about the new portal or any of those new assets or capabilities, feel free to ask as well. And Lisa, I have to ask, going back to your presentation, the GXI, the blood transcriptomic, what would that look like in terms of a recommendation? Is that still related to food? I'm just curious.
Still in progress. They're hoping to launch that test in September. So it's still like a big research and development project, but yeah, it's a big part of what we've been working on lately here.
And everything always takes longer than you think it's going to.
Of course, yeah.
So the question for anybody that doesn't see the chat is around Habit and the updates, because obviously what Habit does very well is meal planning and lifestyle integration. And when would we have the capabilities in Viome to do the same? So it's unclear at this time what that time frame is going to look like. You guys will obviously be the first to know once we get close on that or are close to a reveal. I mean, that ultimately is the biggest benefit of now partnering with Habit. So it would be great to get in there. But the reality is that right now, some of the changes, and you guys will hear about these in the coming weeks, going on with the app, which we're very excited about, we think are going to be just as meaningful for people in terms of their results. So the engineering team has been working furiously for the last couple months to get this summer update out. And then we will we'll see what the future looks like for Habit. So that's a long-winded, very politically correct way of saying I don't have an update at this time, but you guys will certainly be the first to know when we do. And we do recognize that that's the whole reason we partnered with Habit. So I know that there's a big excitement around getting those capabilities married. So I don't think there's any other questions. So at this time, we're going to end early. Want to say thanks to everybody for tuning in. And just a reminder and a thank you to you guys, the ambassadors, for all of the feedback that you provide. People shoot me texts and emails all the time about what they love or what they would want to improve in the customer experience, in the way the results are presented, all these sorts of things. And we're constantly trying to listen to those and then account for them and create changes to the platform and to the app, etc. for you guys. So really appreciate all your guys' feedback. I also want to say thanks to the full team at Viome that puts these things together. Lisa, you were tremendous. Thanks so much for doing this. It's been a long time coming. And thanks to Ted and Erica and Ashley and all of the folks who make this a reality every month. So thanks, everybody. Have a great weekend. And if anybody has any questions after the call, feel free to shoot myself an email or to the support line at Viome. So thanks so much, guys.